I just wanted to start introducing the chair of the second session, Professor Georgia Hodes from the Virginia Polytechnic Institute and State University. And Dr. Hodes' work in mice and human subjects identifies the underlying biology that contributes to vulnerability and resilience to stress and mood disorders, respectively. She's interested in understanding how the sex of an individual contributes to their susceptibility to stress and depression. Her goal is to develop novel personalized treatments and bioassays for mental illness, so these disorders can be medically diagnosed and treated effectively. And currently, she runs her own research lab at Virginia Tech, examining how sex differences in the immune system interact with the brain to drive behavioral differences in susceptibility and resiliency to stress. So before I hand it over to Georgia to introduce the next session, I just wanted to remind the audience to please post the questions in the chat box during the presentation. And at the end of the session as well, we're going to have an open mic and with video on so that we can interact with each other as well. At the end of the second session, we're going to have a short break so that everybody can take a little bit of a rest and before we start. So at the end of this session, there'll be a break. So over to Georgia.
Thank you so much for that introduction. And we're going to be talking now about some more translational work in moving into humans on addiction and sex differences in neuroimaging. And for the sake of time, I'm going to just jump ahead to introducing Dr. Jill Becker. She's the Patricia Y. Gurin Collegiate Professor of Psychology, Research Professor in the Molecular and Behavioral Neuroscience Institute, and Senior Neuroscience Scholar at the University of Michigan, Ann Arbor. And she is a pioneer in this research. Her research over the last 30 years has been investigating how gender and sex and ovarian hormones influence the brain and behavior. We think these findings are incredibly important for our understanding of the underlying neural processes involved in sex differences in drug abuse and in other neurological disorders.
Thank you, Georgia. And I am not leaping over to humans yet, so you will have to bear with another preclinical model study, but I'm delighted to tell you about the work I've been doing over the last 30 or more years, looking at the mechanisms underlying sex differences in addiction. And so I'm going to tell you a little bit about the developmental processes involved in seeing a sex difference in the reward system. Then I'm going to look at talk about how estradiol is enhancing dopamine release in the dorsal striatum and nucleus accumbens. And then we'll talk about the mechanisms through which estradiol is acting by talking about the various multiple estradiol receptor subtypes. And obviously we have a different mechanism as well that Dr. Calipari mentioned. And then we'll talk about how this impacts drug abuse in male and female rats. So as I'm sure everyone here knows, sex differences in the brain emerge as a consequence of both exposure to hormones in the male, where masculinization occurs, and the absence of hormones in the female. And then again, this is during a sensitive period during early development. And then again at puberty, there are permanent alterations that occur. What I've shown is that the absence of hormones during the perinatal period and the presence of ovarian hormones at puberty are necessary to see an effect of estradiol on drug-taking behavior and on the response to estradiol in terms of an enhancement of dopamine release. So I've shown that estradiol enhances dopamine release and that this is a sexually differentiated process, both in vitro and in vivo. So we've shown that you can take the striatum out, put amphetamine on it, and you get enhanced dopamine release if animals were pre-treated with estradiol, or if you add estradiol directly to the superfusion bath, or you can do it with microdialysis. And here, this is with microdialysis after ovariectomy. What you see is that there is an attenuation of amphetamine-induced dopamine release, and you can reinstate the responding to amphetamine with either subcutaneous estradiol benzoate or estradiol administered through the microdialysis probe. So you see there's a rapid enhancement of amphetamine-induced dopamine release in ovariectomized animals in vivo. There are 3 different types of estradiol receptors: ER alpha, ER beta, and metabotropic glutamate receptor 1, or GPR1 as my graduate students call it. ER alpha and ER beta can act at the nucleus, or they can act at the cell membrane, where they bind with metabotropic glutamate receptors to do intracellular signaling, and GPER1 acts is a membrane receptor that mediates rapid effects of estradiol extracellularly and intracellularly. We know that all 3 of these receptors are mediating effects of estradiol in terms of dopamine release and drug-taking behavior. So ER alpha, if you overexpress ER alpha in the dorsal striatum, you can get an enhancement of the attenuation of GABA release that is seen after estradiol. So estradiol attenuates potassium-induced GABA release, and with overexpression of ER alpha, you get an enhancement of this effect. There's a sex difference in the effects of estradiol in cocaine-induced dopamine release in dorsal striatum, where you see that ovariectomized females on the left, castrated males on the right. And what you see is that estradiol enhances dopamine release induced by cocaine in the dorsal striatum in females, but not in males. And we don't see the same effect in the nucleus accumbens. There's a sex difference in that males have a greater cocaine-induced dopamine release, but females are not showing an enhanced dopamine response. If you have a large microdialysis probe that basically is sampling both core and shell in the nucleus accumbens. My graduate student at the time, Katie Yost, didn't believe that there was an effect of estradiol in the nucleus accumbens, in part because of Dr. Calipari's research. And so she did an in vivo study with fast-scan cyclic voltammetry, looking at the estradiol receptor subtypes. And what she saw an enhancement of stimulated dopamine release in the nucleus accumbens shell with estradiol. The ER alpha agonist did not produce the same effect, but the ER beta agonist did. No effect in the males of estradiol or either of the 2 agonists. So ER beta seems to be mediating effects of estradiol to enhance dopamine release in the nucleus accumbens shell. And finally, Chris Turner has recently shown, using fast-scan cyclic voltammetry in freely moving animals, this is a 16-channel probe, he's shown that G1, the GPER1 agonist, enhances stimulated dopamine release in the dorsal striatum of males. There's an effect of GPER1 in females, but it seems to be pulse dependent, which is, duration. A longer pulse actually gets an attenuation of dopamine release. So there are rapid effects of estradiol and dopamine release in females acting in the dorsolateral striatum and the nucleus accumbens. ER alpha, ER beta, and GPER1 all seem to be involved in modulating dopamine release. And GPER1 is also enhancing dopamine release in the dorsal striatum of males. And it's the first time we've seen an effect of an estradiol agonist in males. You saw this figure earlier. So what does it all mean? And you saw earlier, Dr. Nair mentioned this figure. So I'll just tell you that estradiol enhances acquisition of and drug taking in female rats after ovariectomy, but there's no effect of testicular hormones in males. As you saw earlier, there's a sex difference in cocaine taking. This is a choice between cocaine and tasty banana flavor pellets. More females choose cocaine over males after about 7 weeks. and females choose cocaine more rapidly. So this is a population difference in that it seems to be females have a greater prevalence of preference for cocaine over sugar. Interestingly, the G1, the GPER1 agonist, blocks conditioned place preference for cocaine in males, but does not block conditioned place preference for cocaine in females. So again, we're seeing an effect of the GPER1 agonist in males, but not females. On the other hand, if you look at cocaine taking behavior using a progressive ratio schedule, you see that females have a higher motivation for cocaine than males do. And when you add G1 in the dorsolateral striatum, you get an enhancement of motivation for cocaine in females, but no effect in males. So GPER1 is clearly doing something in both males and females, but it's in different directions. So what I've told you is that the mechanisms underlying sex difference in addiction are dependent on the absence of hormones during the perinatal period and the presence of hormones at puberty. Estradiol is enhancing stimulated dopamine release and the acquisition and escalation of drug taking in females. It looks like at estradiol acting at the GPER1 site, receptor is attenuating preference for cocaine in males, but enhancing motivation in females. And the effects of estradiol are being mediated by ER alpha, GPER1, and ER beta in females, while GPER1 seems to be a very interesting target of interest for looking at mechanisms mediating a decrease in drug-taking behavior, especially for males. So these are the people who did all of that amazing work. Cynthia Chestek created the 16-channel fast-scan cyclic photometry electrode, and thank you.
Thank you so much. Perfect timing. In fact, you even had like 50 seconds to go. I'm so impressed by your punctuality. I'm going to move on to Professor Rita Z. Goldstein from Mount Sinai. She is a Mount Sinai professor of neuroimaging and addiction in the departments of psychiatry and neuroscience at the Icahn School of Medicine at Mount Sinai. Dr. Goldstein is the chief of neuropsychoimaging of addiction and related conditions research group. An important application of Dr. Goldstein's research is to facilitate the development of intervention modalities that would improve cognitive and emotional function, leading to better treatment outcomes in drug addiction and other chronically relapsing disorders of self-regulation.
Okay, can you see my screen, the slides? Can you hear me?
Yes, we can hear
yes.
you. We can see your next slide though.
Oh. I'm sorry, just a minute.
Still can see it.
Okay, just a second. How about now? And then let me just bring up this.
Yes, perfect.
Okay, okay. So thank you very much for inviting me to present in this very important symposium. I'd like to start by acknowledging that it takes a whole village to take this type of studies in humans. And especially I'm going to highlight the work of Yuefeng Wang, postdoc in the lab, and our funding, mostly from NIH and from NIDA, so thank you. I also want to start with describing our IRISA model, where we emphasize that impairments in response inhibition and salience attribution are core to understanding the basic clinical symptomatology of addiction, as you can see here. So expanding the view beyond the subcortical involvement in reward and habit to the prefrontal cortical roles in self-control and regulation and salience attribution, with the hypothesis that drugs assume a heightened salience and value at the expense of all other salient cues and reinforcers, and with a concomitant decrease in the ability to inhibit behavior. So this is just the prefrontal cortex focus. And when we reviewed the literature, and this is with Anna Zilverstein, she's going to be talking in the next session, but this is when she was a postdoc in the lab, we reviewed the fMRI studies that compared people with addiction to people without addiction, and showing that across the board, if you expose people with addiction to drug-related cues while they're in the scanner, these are fMRI Bold studies, You have activations not only in reward and habit areas, but also in other brain regions including the salience regions and higher order executive function regions that have to do with this self-control and self-regulation. So this is an example, and this is a study we published where we show, so when I talk about this type of studies, usually people show drug-related cues, pictures, while a person is in the scanner. So those are drug images like what you see in the middle. And also we show them neutral type of images, but also other salient images, for example, food cues. And here in people with heroin use disorder, as compared to healthy control subjects, when you compare drug-related cues to neutral cues, you have this fMRI bold activation. Here is an example in the right orbitofrontal cortex. And the higher this drug cue reactivity, the more the subjective perception of craving. So it's a marker of cue reactivity and the perception of craving. And we saw this type of pattern also in the ventromedial PFC, but also in the ventral striatum and in inhibitory control regions like the inferior frontal gyrus, even when comparing those drug cues to the salient food cues. But in our study, here most were in the healthy, both groups, most are men. So it's 62% of individuals in the control group that were men, and almost 80% of people with the heroin use disorder were men too. And this reflects actually the state of the field. This is again back to Anna Zilberstein's paper where we show that out of these more than 100 of papers, and most of them did not include any women or very few women, and only 7 out of more than 100 studies were powered enough to conduct any sex differences analysis. So, and it's not only, does not affect this underrepresentation of women in our type of studies, does not affect only clinical neuroimaging, but also in general, also clinical trials. So we think, based on animal studies that we just heard, that women show greater severity of drug abuse, faster progression to problematic drug use, and higher relapse risk. But there are also many opposite results. And when we reviewed the epidemiological type of data, we showed that if anything, men have much more propensity than women for overdose mortality across different drugs of abuse, across all United States, across the ages. So we were lucky to receive funding to study sex differences and menstrual cycle effects in humans. And it took us only 10 years and 14 submissions to get this funding. So here I'm going to present results of a study where we included this time 49 men with heroin use disorder and 32 women with, half of them were with heroin use disorder and half with cocaine use disorder. There is still an issue of recruitment, so we have to combine groups. Most of them are white. They're in their late 30s and early 40s. They have high school level of education, average intellectual functioning. Most of them are cigarette smokers. They have mild depression and anxiety levels and moderate stress, perceived stress. They have about 6 months, average of 6 months of abstinence when we see them. In the men, they're inpatients, so they have no drug use in the last 30 days. Women have higher past 30 days drug use, especially with cocaine, but this difference did not contribute to any of the results I'm going to show you. They started drug use at their early 20s, and they've been using the drugs for over a decade. We recruit them through treatment facilities and organizations like the Samaritan Daytop Village and the surrounding community. And in a subset of the women, in half of them, we ran the study twice. So in the follicular phase and the luteal phase, and we randomized the first scan based on the follicular versus the luteal. Okay, so what are our results? We see that when compared, this is fMRI bold in the medial PFC, when people are in the scanner just passively looking at those images. And when you compare the drug images to the neutral images, you have hyperactivation in this region for women. It's the same pattern of results. If you compare the drug to the other salient cues, which are food, you have this drug reactivity. In contrast, in the dorsolateral prefrontal cortex and inhibitory control region, you have reduced drug reappraisal as compared to men. And reappraisal is a way to down-regulate your emotion, to down-regulate your reactivity. We ask people to look at the picture, but now think about how it's only a picture, it's only a movie prop, it's not your drug, you cannot do it. So really reinterpret the picture, not just distance yourself from it. So men reappraise in this region much more than women, and the more their reappraisal, the lower the cue-induced drug craving. So as if they're mobilizing this control region to reappraise those drug cues, to reduce drug reactivity and regulate their cue-induced craving. What happens during menstrual cycles? So here is a subgroup of the women who we scanned the follicular versus the luteal phase, and we can see this drug cue reactivity in the dorsolateral prefrontal cortex, in the follicular as compared to the luteal phase, in the dorsolateral PFC. In another control region, the inferior frontal gyrus, the more this drug cue reactivity, specifically in the follicular as compared to the luteal phase, so this is delta, the more it is, the lower the the higher the cue-induced drug-related craving. So the more the reactivity, the more the craving, specifically for the follicular, more than the luteal phase. And the VMPFC shows a very similar pattern. The more the reactivity, specifically for the follicular, the luteal phase, the more the craving. And it is associated directly with estradiol that we measured in blood samples in the same day of the scan. So such that the higher the drug cue reactivity, as compared now directly to the food cues, the more the estradiol in the follicular, the luteal phase. And what happens with the luteal phase? We have the opposite. So in the luteal phase, we have reappraisal higher than in the follicular phase. And the more this type of reappraisal activity during the luteal phase, the more the progesterone as compared to the estradiol directly. In the VMPFC, again, it's the opposite happens with estradiol. The more the reappraisal in this region, the lower the estradiol specifically for this luteal versus follicular phase, and a pattern that we see also in the anterior prefrontal cortex. So to summarize, we saw sex differences such that women more than men show higher drug cue activity, lower drug reappraisal. We also see menstrual phase differences such that the follicular more than luteal phase shows more drug cue activity associated with more craving, reduced drug reappraisal, and it's associated directly with hormones such as estradiol and also progesterone that's associated with more drug reappraisal. And to conclude, the follicular, it's very consistent with the animal models. The more the follicular phase and estradiol are risk factors associated with higher drug cue activity, reduced reappraisal. The luteal and progesterone are protective. It's more drug reappraisal in frontoparietal control networks. So maybe when we're designing interventions, we have to reduce drug cue activity, increase reappraisal, and other control functions, such as alternative reward savoring, during specifically the follicular phase. And I want to end by thanking Nelly Klein, the co-chief of the lab, my partner in crime for 2 decades, for across our cycles. Thank you.
Thank you so much. That's fantastic. Moving right along to Dr. Christina Dalla. Dr. Dalla will have a recorded talk, but she will be here to speak during the question and answer session. Dr. Dalla is a professor of pharmacology at the Medical School and Second Department of Obstetrics and Gynecology at Aretaiao Hospital of the National and Kapodistrian University of Athens. She is president of the Mediterranean Neuroscience Society and president of the Hellenic Brain Council. And her work focuses on sex differences in neuropsychiatric disorders and novel treatments with a focus on depression and anxiety.
Hello, thank you very much for the invitation to this very interesting and very important meeting on sex differences. So as we know, there are sex differences in neuropsychiatric disorders. Here you can see some global data on the daily disability adjusted life years for men and women, and it's also based on age. So on the y-axis you can see the years, and we can see that females suffer more from depression and anxiety with red, especially after puberty. Also, many of these disorders comorbid with addiction, and this is very important to understand why it's happening. Also, we know that there are sex differences in the brain at all levels. There are sex differences in the expression of proteins and genes. And here we can see the percentages for many different disorders, but also for brain regions in a very large sample from a very big study that was very recently published. Of course, we also know that there are sex differences in drug response, in pharmacokinetics and pharmacodynamics of psychotropic drugs, adverse effects and also efficacy. But we even recently have found that there are sex differences in the blood-brain barrier transport of psychotropic drugs, something that is not very well known so far. And for this, we advocate that we really need to integrate sex and gender in mental health research and in general in psychopharmacology research. And in order to do that, especially for clinical studies, but also for preclinical studies, we need to enhance funding and have more targeted studies on sex differences. So this is something that has also been incorporated in European research. The European Commission has issued guidelines about why integrating sex and gender and intersectional analysis into research. And there are many information on how we can do that, and also motivation to do it is also very important. But what about preclinical studies that we're usually doing in our laboratories? We have repeatedly argued in the past, also with our excellent colleagues that they're also here today, Professor Deborah Bankhasser and Professor Georgia Hodes, that we needed to do studies in both male and female animals because many times in the past there were studies that they were done only on male animals, and then many compounds failed when they entered clinical trials because they were mainly tested on women. So there was a mismatch between preclinical and clinical studies, and what we argue is that we need to do research on both males and females. We'll see later also how to have a good design on that, and then if there are sex differences, to stratify by sex and have human studies, clinical trials that they can really detect sex differences. This is also something that we have written in the past at Nature Reviews Drug Discovery with our colleagues from the Women's Brain Project and the European Medicines Agency, that they also understand that we need to really implicate sex-related differences in the central nervous system in drug research and development. So in our laboratory, we have the last decades worked on mainly on animal models of depression, anxiety, cognition, and others, but also on drug response and psychotropic drug response, as you can see here. I want to show you much data. I just want to show you one example of some recent studies that we have done recently with my excellent PhD student, Pavlina Pavlidis. These are studies that they are ongoing, and I just want to highlight this as an example of how sex differences in drug response can be important. So we study the GPR1 neuroestrogen membrane receptor. It's a receptor in the brain of males and females. It's not a classical estrogen receptor, but a membrane receptor. And we investigate whether this could be a target for rapid-acting treatments. And in order to do that, we compare these compounds, agonists and antagonists of this GPR1 estrogen membrane receptor, with classical treatments on male and female adult rats. Of course, on females, we always do vaginal smears for the estrocycle, and we do the behavioral test and acute studies in order to compare and see if there is an effect of the G1, which is the GPR1 agonist, and or G15, if this effect can be blocked by the antagonist, G15, or if there is an effect that is similar to fluoxetine and ketamine. As you can see, these are large studies with many animals. It's important to have a statistical power and have an experimental design that can reveal the sex differences, and also to have the different combinations between the pharmacological treatments. Just a few results to show you the different effect of ketamine on females and males on the novelty suppressed feeding test. This is a rapid effect where ketamine shows a decrease in the latency to approach the novel food. So at this behavioral test, we have animals that are food deprived, and then we measure how much time they need to go and approach the food. But as you can see, there are also sex differences at baseline, and females approach the food much faster, female rats, than males. And this is a very nice example of how a treatment at this example, ketamine, can have a differential effect on males and females. Also, on the neurochemical level, we have seen on the same animals that G1, which is the agonist of the GPR1 receptor, the estrogen membrane receptor that activates this receptor in the brain, increases dopaminergic activity at the prefrontal cortex, as we can see with the ratio of the metabolite of dopamine HPA to dopamine, and this is reversed by ketamine, but this is only happening in females. Of course, we have many other different data from previous studies on stress response, which are similar, and also effects of drugs, and many other labs have investigated this, and we have all seen differences, sex differences in drug response that they really need to be investigated and understood. For the experimental designs, the astrocycle monitor, and also for statistics, you can find this recent paper on methods, Journal of Neuroscience Methods. It's for practical solutions for including sex as a biological variable in preclinical neuropsychopharmacological research. And there you can also read that many times statistics and design are the most important methodological issues, but of course, how we can do studies with gonadal hormones in both males and females. This paper, as you can see, it's written with many experts that you probably know most of them. It's based on a series of open access videos that you can find on this link. They are available online openly, and they have different modules that we explain all the methodological issues on for preclinical and basic research with males and female animals, mainly for mice and rats, but also there are some implications for clinical studies, for human studies, and even for surgical approaches, behavioral, and molecular, and others. I hope these are useful and they can really contribute to the enhancement of this field. And so with this, I would like to thank very much my lab and my collaborators, especially our funding sources, our collaborators in Greece and abroad, and my colleague, Assistant Professor Nicholas Kokras, who has really contributed in these studies, and we have worked together for many years. And also, lastly, I would like to invite you all if you manage to be there next year at Crete for the Mediterranean Neuroscience Society meeting, where I have the honor to be the president, and we'll have a conference next year at Hania in June. So thank you very much.
Thank you. And if everyone could, if people are comfortable with turning on their cameras for the discussion, we would appreciate that. We're going to have 10 minutes for discussion, but we are going to skip the break just to catch up on time. And I'm going to start immediately with a question from Dr. Valentino to Dr. Becker. Do we know the molecular mechanisms by which estradiol affects dopamine release, and what would it be different in males and females?
So the model that we're working on is that estradiol is decreasing GABA release in medium spiny neurons, and we've shown that estradiol decreases GABA release, and I showed you a bit of that, and that it's estradiol is acting by metabotropic glutamate receptors on those GABA neurons, and if we block the metabotropic glutamate receptors with MPEP, we block mGluR5, we prevent the effects of estradiol to enhance dopamine release. So that's the model that we're working on.
And
Jill, I guess I was just wondering, is it somehow interacting with the receptor, or?
Well, I think it's a presynaptic GABA. I think that GABA is inhibiting dopamine release presynaptically.
Right.
So
but
it's
how
right
is
on the
estradiol affecting the GABA then? I'm trying to back up and figure out the molecular.
Yeah, so I think my ER alpha associates with mGluR5. And so it's an intracellular signaling system. We can prevent the effects of estradiol by blocking mGluR5.
Okay, thank you.
I have my own question for Dr. Becker, which is, are you seeing the different types of estrogen receptors on the same cells, or are there specific cell types that you're seeing ER alpha versus beta versus GPER on?
That's a terrific question. And one thing, I have a graduate student who's really keen to be looking at that. So I hope to have an answer for you one day soon.
Excellent. I'm going to switch to Dr. Goldstein. And for Mariana, we have a question. Could you please explain those why those differences are observed between the follicular and luteal phase? I would have thought that there would be more negative affect during luteal phase and therefore decreased capacity for reappraisal.
Yeah, so what we see are in the follicular phase and correlations with that has higher estradiol, we see correlations with more drug craving. In the luteal phase, where we have more progesterone and more progesterone over estradiol, we see correlations with food craving, actually, that I didn't mention. So I think it comes along, and maybe it is part of the story. And the fact that the luteal phase actually may be associated with more reappraisal and more control is also consistent with the study by Teresa Franklin that showed that if women with nicotine dependence, if they start treatment in the luteal phase, they have more chances and better success rates at stopping nicotine smoking. So I don't know yet the answer to that, and we definitely need to look at the affect type of differences within the follicular and luteal phase, but the correlations were extremely consistent that during the follicular phase, there is more craving and less regulation of craving with an opposite pattern in the luteal phase that we definitely have to explore a little bit more. And the food savoring is another way to actually reduce drug craving because you are upregulating alternative reward processing. And so that was associated, that is another way to maybe reduce type of craving. And again, I think we saw that that actually in the follicular, Yuefeng Wang, who conducted those results, he can also pitch in a little bit, that may be a way where the follicular phase will be involved in another way to regulate craving.
So then would it make sense
Awesome.
to
Very
put
interesting.
women on, oh, sorry, go ahead.
No, I was just going to say thank you. That was very interesting. Thanks. Very interesting findings.
Would it make sense then to be putting women on contraceptives to help with getting rid of the cycle, basically?
Oh, That's a very interesting question. I definitely would want people to know that the follicular phase is a little riskier based on these fMRI studies. We have to link them to behavior. The only link here is through craving. And also the reappraisal, it's only through fMRI both. I still would like to explore actual behaviors and emotions before I would make any more type of conclusions that are broader based. But it's clear that maybe that there is protection during the luteal phase, again, specifically against craving and to reduce craving for people with addiction, severe type of addiction. So I wouldn't make any more general statements than this right now.
Sure. And Dr. Becker would like to know, have you looked at estradiol progesterone ratios during the luteal phase? Do you think it's necessary to have estradiol priming to see the effect in the luteal phase?
So the priming I'm not sure about, but we measured in every woman their estradiol, progesterone, and the progesterone over estradiol calculated ratio. We have it during the follicular and luteal phase. As expected, the estradiol is a little higher during the follicular than luteal phase, but it's only a trend because we're measuring here the mid-follicular and not early follicular phase. In early, you would see the reductions in both, so we don't have that. We have increases in both estradiol in both the follicular and luteal phase. In the luteal phase, we have clearly very strong differences with progesterone, which is much higher than in the follicular phase. And also the PE ratio is much higher in the luteal than follicular phase. And what I like about this analysis is that every woman is her own control. So we have for every woman scans at both phases. And then all the myriad of variability in humans with addiction is controlled for.
Excellent. Dr. Dalla, Dr. Becker wants to know if the effects of on the effects of G1 and ketamine are really interesting, and she wanted to know how you administered the G1 and at what dose, and how do you think those drugs are interacting?
Yes, thank you for the question. And I found your data very interesting, and we should talk more, hopefully at CRET in Greece. We administered G1 with IP injections. It's a dose of 0.01 milligram per kilogram, and it's 10 minutes before the behavioral tests. And we think we're actually investigating now there are probably some common pathways between ketamine action and GPR1 activation. So we have some data that maybe some pathways like the ERK or the mTOR, they are synergistically in a way involved in these effects, but we'll know more next year. And for the next, I think there is another question for the what behavior is the novelty suppressed feeding test used to model? We use it for, it's mostly about emotionality, anxiety, and some depressive symptoms. It's not addiction.
I have a question for Dr. Dalla. Given that estrogen receptors are really promiscuous, and a lot of the talks that we've seen have also been about how estrogen may also act in different ways, particularly the GPER, more recently discovered GPERs and these fast-acting effects of estrogen. Do you think that we should be moving more in that direction to understand how estrogen is binding to things other than estrogen receptors?
Yes, I think so. And also to differentiate the rapid effects from the slow classical effects, and see, for of course, the sex differences, because we saw that they can have different effects in males and females. And also many times they are totally opposite effects, and also they depend on the if the rats are ovariectomized or castrated. So yes, we need more research about all these aspects. Thank you for the question.
Welcome. We have 1 minute left if anyone has a question that they want to ask. If not, then please join me in thanking our speakers.